Thank you very much, Edith. And first off, I'd like to thank ILSI and ILSI staff for not only funding, but providing us a lot of help during the initial organization of this project. And you'll see in a little while, it was quite a task to organize some parts of the project. So the project I'll be speaking on is looking at Listeria monocytogenes. Some of my other colleagues, as Edith mentioned, will look at Salmonella and viruses. So what I'll do in the next 25 minutes or so, give you a brief introduction to a few different areas, to LM, low moisture foods, and who was involved in the ILSI study. Then go on a little bit to look at some of the recalls that have been associated with Listeria monocytogenes, and then get into some of the work that we have done so far in the particular study. So I'm not going to spend too much time on this. Everyone has been, hundreds of talks on Listeria and the symposium, so I'm not going to go over this. So, but I did want to go over the team members, and I'll go over some of the other team members who I'll acknowledge at the end of this talk. But the main co-investigators who work with me, Dr. Saba Badawit from Health Canada, who will be speaking after me, Dr. Sophie Cathario as well from North Carolina State University, who will also be speaking, and Dr. Keith Warner, a colleague of mine from the University of Guelph, Canada. And these have been excellent collaborators with me on this project. I think we had some difficulties at the start because we tried to standardize everything. So you can imagine shipping things like cornflakes or dried fruit across the border, even to from Guelph to Ottawa. So we tried to get things standardized, and took us a little longer than we thought. But we've also, I think, learned from each other. The groups, I think, have learned from each other as we went along. But it was a lot more complex than I originally thought, working with the labs. Even Dr. Warner works in a different lab from me. So we had to have him come over, and with special MTA agreements to carry strains, which, as we know, some of the safety issues involved, we need to do that very carefully. As most of you know, low moisture foods are those with a water activity below 0.85. There's a very, very wide range of low moisture foods that are out there. And historically, even when I started off in Health Canada, we never paid attention at all to low moisture foods, not even on our food safety questionnaires during outbreaks. But we now know that since a number of outbreaks linked to low moisture foods, but although in the main part linked in the main to Salmonella and staph, there have been a number of other organisms as well that have been involved in outbreaks linked to low moisture foods. So definitely a concern, and there needs to be more worldwide recognition of this concern, not only in North America, but throughout the globe. And I guess the concern with Listeria monocytogenes has been that not so much we know that Listeria monocytogenes cannot grow in these low moisture foods, but as you'll see from some of our studies and other studies, it can survive fairly well on these products, especially when stored at 4 degrees C. So basically, where those countries that have a zero tolerance for Listeria monocytogenes, if they find Listeria monocytogenes in a ready-to-eat low moisture food, it's going to be recalled, even though the food cannot support growth of the organism. And we know, for example, there have been outbreaks linked to ice cream, outbreaks linked to peaches, where very low numbers of the organism were found. It doesn't mean that in all cases very low numbers cause illness. But I think as Listeria people, we're starting to realize that even very low numbers of organisms can cause disease in exquisitely sensitive individuals. Okay, so some of you may have seen there have been quite a number of recalls of Listeria monocytogenes due to its presence in low moisture foods. I've just listed here some of them. for the most part here. We've listed the nuts and peanut butter and the like, but you may have seen even, I think it was either yesterday or today, raw pet food was being recalled for Listeria monocytogenes. Some of the studies that have been going on, there have been definitely some studies looking at the survival of Listeria monocytogenes on low moisture foods. So you can see a whole host of here, again, raw almonds, pecans, pistachios, walnuts. And for the most part, as an overview of these studies, Listeria has been able to survive fairly well at low temperatures. And at higher temperatures, room temperature, 22 or 23, you see a slow decline with time. So that's sort of the picture, some of the picture that's emerged with some of the products that have already been studied. So in terms of the foods, this is the foods that the ILSI North American Committee wanted us to take a look at. And so you can see here on the top some of the dried fruit products, raisins, strawberries and apples, and also chocolate liqueur, pistachios, and cornflakes. So quite a wide variety of products that were looked at, examined in this particular study. So getting into some of the objectives in our particular study, a number of things came out as we started into the study. One is that the ILSI committee wanted us to use dry inoculation for the dried fruits. So you'll see we have dry inoculation for the 3 dried fruits, and as you'll see later on, we use wet inoculation for the other 3 products. We also wanted to take a look at, as some of you have seen in the literature, there are a number of dry carriers, and there's still some discrepancy about what's the best way of doing dry inoculation. So we tried to take a look at some of the ones that were out there most people had used. Also looking at the survival and potential changes of virulence of Listeria monocytogenes as it's sitting on these low moisture foods for periods of up to 1 year. So is there anything, are there any changes, can there be any changes in the virulence of Listeria monocytogenes over this long period of time? Also, we wanted to take a look at the microbiome of the low moisture foods during the storage to see if we can do any correlation between how LM is acting, surviving, or its virulence, and the microbiome of the low moisture foods. So I'll go over just briefly some of the methodology, and we'll go over some of the methodology for the latter part of the study in a few slides as well. So as you can see here, again, we have the dry inoculation on left-hand side with the dried fruits, looking at the talc, sand, and chalk as potentially what would be the best carrier. Then we did the wet inoculation on pistachios, cornflakes, and chocolate liqueur. And we used a few different means of inoculation. And my master's student, Vivian, did that work and looked at a few different means of inoculation. The incubation period then was done, incubation was done both at 23 C and 4 degrees C. In the sampling, we are going to do over a 12-month period. And unfortunately, we're not at the end of the study right now, but it's in progress, and you'll see some of the results that we have gotten to date. In this particular study, we used a cocktail of Listeria monocytogenes. We wanted to use a variety of different Listeria monocytogenes serotypes. So we used the 1/2 A, 1/2 B, 3A, and 4B as our serotypes in this project. Okay, so getting on firstly with the wet versus the dry inoculation. For the wet inoculation for the chocolate liqueur and cornflakes, Vivian found that misting turned out to be the best for these particular products, and I'll show you a picture of the mister that we did use. For the pistachios, we used a massaging of the liquid for the pistachio so we can get it actually inside the pistachios as well as outside on the shell. In terms of looking at the dry inoculation, the best overall that we found was the sand, actually. So the sand quickly came back to equilibrium, back down to the AW that we started with before we put the liquid on. It was easier than the others to spread and easier to work with, and it also gave overall better recovery of Listeria monocytogenes. So didn't have time, obviously, to show all the results. We did a lot of experiments in this particular area, but here you can see, looking at the sand versus the talc. Sorry, the pointer is just not working here. But the sand, as you can see on the top there, giving better recovery there in some of our preliminary experiments. And the talc as well gave quite a large loss initially, 2.8 to 3.3 logs initially upon inoculation. So taking everything into account, we decided to use the sand, although the sand itself, as many know, is still not the panacea, and there's still issues with using dry carriers. So starting off, what are some of the results that we got looking at the survival of Listeria monocytogenes? This was survival looking at the survival of Listeria on the dried apples. And a little surprisingly to us, we were unable to recover or enumerate Listeria monocytogenes from any of the dried apples. The apples did not have any sulfites, also had a fairly low pH, but you'll see later on some of the other products also had a low pH where Listeria did survive. So potentially something else going on there on the dried apples. This was the picture on the dried raisins up to where we are now, about 180 days. You can see Listeria surviving fairly well at 4 degrees C, and quite a sharp drop off at 23 degrees C. And we went basically looking at where we had 3 consecutive negatives, which is what sort of the ILSI committee and ourselves had agreed on. We'd be stopping sampling after having 3 consecutive negatives. A similar picture on the dried strawberries. pH here was about 3.9 on the dried strawberries. So you can see there was a little more of a tail off there at 4 degrees, but still surviving. And again, very sharp tail off at 23 degrees C, and then negatives throughout after that period of time. Okay, this is looking at some of the wet inoculation. This was the retail pistachios that we used. And you can see there at 23 degrees, doing fairly well, almost as well as the product stored at 4 degrees C, the only one where we saw fairly good recovery at 23 degrees Celsius. And here you can see there, that's the mister that Vivian used in her studies. This was the cornflakes, again, somewhat similar in that we've had fairly good survival so far at 4 degrees C, and we've got a slow tailing off at 23 degrees, but we're still being able to recover it after 160 days. This is the chocolate liqueur. Again, 4 degrees, more or less stable after about 80 days. And again, we see a slow decrease at 23 degrees at the chocolate liqueur, but still surviving and hanging in there at 23 degrees. Okay, so what we're also planning to do during this study, because we had the 4 different serotypes, we're planning to look at, is there one serotype, for example, that is able to survive better on these low moisture foods? So I think that's very important. Not too many people have looked at the differences in serotype survival. So Vivian was able to actually develop a multiplex PCR for serotype differentiation of these 4 different serotypes. And here you can see the results of some of the results that we got out, very clear banding patterns, and we're able to get distinct banding patterns for each of the 4 different serotypes of Listeria monocytogenes. So we're looking forward to getting some of those results coming out. In terms of the microbiome, just very quickly, this is some of the methods that we're using. This is the isolating culturable methods of the low moisture food microbiome, where we're looking at both growth, where we're streaking isolates with different phenotypes out and then going to be sequencing those, where we have no growth, enriching the foods in BHI broth and resuspending, and then taking a look at doing sequencing and coming out with what we find as a microbiome on those low moisture food products. This is just some of the results that we've got so far on the culturable methods of the low moisture food microbiome. In the most part, I would say not a heavy background microflora on the 6 low moisture foods that we looked at, but you can see the raisins and the dried roasted pistachios actually came out with the most number of different phenotypes, and we'll be working to identify those as we move along in the study. So in terms of some of the future work that we're going to be doing, we're going to be continuing on, obviously, to the 1-month period for those low moisture food products where we still see survival of Listeria monocytogenes. For the low moisture food microbiome characterization, we're also going to be doing culture independent work, looking at the microbiome where we do direct DNA extraction and sequencing, and then going on to the MiSeq. As I mentioned, we're also going to be doing the serovar tracking, looking at the multiplex PCR. In terms of the virulence potential of Listeria monocytogenes, so what we're going to do is take strains at time 0, and then at the end where we see the latest survival of Listeria monocytogenes, and then doing different virulence assays of those Listeria monocytogenes strains that we get out to see, for example, have there been any changes between the time 0, day 0 on the low moisture food versus 3 months after it's been stored, let's say, on a dried fruit product. So for right now, we are planning to do both the in vitro work with looking at adhesion and invasion in CACO2 cells, and we're also going to be doing the Listeria model, in vivo rate of killing. So we've been working very closely, Sophia and Victor. They've been extremely helpful to us. They've been working for quite a long time with the Salmonella Listeria model, as you'll see in a little while. And Victor has been very helpful. He even took videos for us and sent the videos to our lab. So that's what I was talking about, the collaboration, where we've been able to work with each other to help each other out as we had different issues. We are going to have to, we had some discussions at lunch actually, we are going to have to potentially think very carefully, because Victor told us that with the Listeria model, it's very, very sensitive, so you need to be in a range of about 10 to the 6 organisms per gram for the assay to work well. So we're going to have to look back and see how we may approach this in terms of there may be some time points where we're going to be below that 10 to the 6. And so we're going to have to, think carefully before we actually progress in this work and see what we may have to do in that particular respect. So in conclusions, we found that sand was determined to be the best carrier for recovery and enumeration of Listeria monocytogenes from dried fruits. Listeria levels declined significantly on cornflakes, dry roasted pistachios, and chocolate liqueur stored at 23. The populations of Listeria LM remained stable on the pistachios, cornflakes, and chocolate liqueur, however, stored at 4 degrees C for 3 to 6 months. Listeria remained stable on raisins, decreased about 1.5 logs in strawberries, and did not survive on the dried apples stored at 4. We were able to develop a new multiplex PCR method, which we'll be using to track and differentiate between the 4 LM serotypes that we used in our inoculum. The raisins and pistachios appear to contain the most diverse microbiomes, each with 10 different phenotypes. One other thing that I'll mention is that some of the strains, for example, if we see differences in virulence between, let's say, day 0, day 3 months, and maybe further on, we're going to be doing a whole genome sequencing on those isolates to see what's going on in terms of potential gene changes on those particular strains. So I think that'll be also an important component. And we're going to be working, we discussed also at lunch with Sophia, possibly looking at some RNA studies, looking at RNA gene transcription for Listeria monocytogenes, looking at survival with time and how does that change the RNA transcripts. So I'd like to thank our whole team. Firstly, University of Guelph, as I mentioned, Vivian Lee, our MSc student who's worked on 3 of the low moisture foods. Ryan Kuzi was another of our master's students who worked on the dried fruits. Dr. Valeria Pereira, who's my research manager, who helped us, has been helping us a lot. Fernanda, who's here in the audience, she was involved in our initial work and helped draw up some of the initial plans with a large number of spreadsheets. Tried to get that into something that we can actually start working with. My colleague, Dr. Keith Warner at Health Canada. Besides Saba, we had Natalie Corneau, Dr. Neda Nashiro, who did also some excellent work. North Carolina State University, Sophia, and her master's student, Victor, who've been doing some excellent work and also have been excellent collaborators with us. And of course, to thank, a big thanks to ILSI North America for funding this large study for us. So I think I'll stop at that point, and thank you very much for coming today.
Thank you, Dr. Farber. We have time for a couple of questions. So if you would come up to the microphone and then announce your name and your affiliation. Thank you.
Thank you very much, Dr. Jeffrey, for the excellent presentation. I'm from Champion Pet Foods, Edmonton, Canada. My name is Gayan. So I'm interested to know that when you did this study, what was the water activity levels at the dried products? I know it's less than 0.85, but was it far lower than that? And then also a follow-up question on. So it's interestingly that at 23 centigrade, in dry inoculum, there was a drastic log reduction, and then at wet inoculum, you didn't see that. So do you have a tentative explanation why that difference is? Yeah.
Thanks for the question. Yeah, the water activities that we used were, I didn't have time to show it all across, but it was much lower than in the range of, let's say, 0.2, 0.3. And so what we did is actually we had to have a drying period. So we had a period of, depending on the type of foods, it was either dried first at 30 or 37 degrees for 24 hours. And then we went down to get an equilibration to make sure it remained stable. So we went down to about 22 to 23 degrees for about 3 days in order for it to get back to the initial AW before we actually inoculated the product. And I think the question that you mentioned about the decrease, I think definitely there were issues with the sand. There are a number of issues when you're using dry inocula. And I think we lost quite a bit of cells when we actually not only inoculating the sand with Listeria, but also when we inoculated I know it gets confusing, the Listeria inoculated sand inoculated onto the low moisture food, we also lost. So there were two different steps there where we lost, where we didn't have those two different steps in the other products. So, but I think, it's something that I think we need to discuss, because I know actually Victor and Sophia brought this up in terms of, and the problem is, you have the sand on the product, right? So you don't know, in a lot of cases, are you actually looking at Listeria survival on the sand? Because you do have the small, even though we sieved the sand out as much as we could, you still have some particles there. So are you looking at Listeria survival actually on those sand particles? Or are you actually looking at Listeria survival on the dried fruit? Or in our case, I think we're looking at sort of a compilation of the both. But I think, as I mentioned, there are issues that I think as long as you point those out, those are some of the limitations. But I think overall, I think we were happy with the results we got.
Since no one else, could I ask a couple more questions? Sure. Okay. Then the other question is, so when you did this study, do you have any recommendation for the industry? Because industry can't use pathogens for their validation stuff. So any surrogate, like, do you see these kind of features, or have you seen any of these kind of features in Listeria inocula, or any kind of surrogate organisms? Or do you have any recommendation for the industry to use any surrogate to see these features in the food matrices?
Yeah, no, it's a good question. Obviously, surrogates is very important. I guess being sort of a Listeria person, I always tend to go to Listeria inocula. But I know there are issues. I mean, obviously, there's many different types of inocula, and some of them don't simulate LM that well. But I think what we do is we try to pick out a range of different inoculas and then see how they behave in common with our Listeria mix. But of course, Enterococcus, and there have been a number of other surrogates that people try to use, especially for Salmonella. But I would still go with Listeria inocula.
Thanks, Jeff, for the presentation. I was wondering, you showed the figures with the error bars. And can you share how you designed the experiment with respect to the reproduction? So, of the inoculum, the product used for the experiments, how were the reproductions done to get these inactivation curves?
Yeah, I mean, Vivian will probably know better than I do, but we had basically, it was duplicate experiments with triplicates, or triplicate experiments with duplicates. I forget exactly.
Mm-hmm.
Thank you for your question. So for our study here, we did the 2 biological duplicates, and then we're sampling in technical triplicates.
And did you also then use different product batches? So, for example, you bought 2 types of dried strawberries, or was it just one batch, which you split it in 2?
Yeah.
Okay.
I'll just go ahead.
I just have one question. Yeah, so we, like you were saying, the single batch split in 2 for our cornflakes as well as the chocolate liquor. But for the pistachios, where, as Dr. Farber presented, there was a presence of a microbiome, we were interested in that aspect. So we have 2 separate lots of dry roasted pistachios that we're looking at the survival as well as the virulence in the microbiome.
So I'll just go ahead. For some of them, as I mentioned at the beginning, we tried to get huge amounts of product. So for the chocolate liqueur, ILSI kindly supplied us. They supplied us with the cornflakes large, because I tried to get one sort of lot that I could ship to North Carolina, and I could ship to Ottawa as well. So when we could, I tried to get sort of one batch representative for the whole study. And the same with the dried fruits. Those we had to actually order from, it's called Vance Foods. They're one of the largest suppliers of dried fruits. Again, we tried to buy huge amounts at the beginning, so we'd have enough, so everyone could have enough to inoculate and last the 12 months.